Our somatic cells are diploid. They contain two copies of each chromosome. One comes from the mother, the other from the father. These chromosomes have two chromatids, as their DNA content duplicates before the cell divides. The DNA content of the two chromatids is identical. When a diploid cell is divided by mitosis, the resulting daughter cells are also diploid, but their chromosomes have only one chromatid. These chromatids break up, and the DNA duplicates before the next mitosis. The chromosomes in the cell will have two chromatids, and cell division starts again. During the process, the number of chromosomes does not decrease, unlike in meiosis, during which haploid gametes are formed from diploid cells. Mitosis consists of four phases. The first phase is the prophase. Chromosomes are formed from the chromatin, which consists of DNA and proteins. The centrosome duplicates and moves towards the opposite poles of the cell. In the metaphase, the nuclear membrane breaks down. The spindle apparatus is formed. Then the chromosomes line up along the equatorial plane of the cell. In the anaphase, chromosomes split into chromatids, forming chromosomes with one chromatid, which then move towards the opposite poles of the cell. In the telophase, the cell splits, and the nuclear membrane is formed again. Then, the chromosomes break up. That is, during mitosis, two diploid cells are formed from one diploid cell. Certain cells in our bodies, such as nerve cells, are not able to divide. Others may be able to divide several times during our lives. The cell cycle is a finely tuned process. Disturbances may lead to uncontrolled cell growth and the formation of tumors.